Polymerase chain reaction. PCR is a technique that allows researchers to quickly create many copies of a specific region of DNA in vitro. It's developed by Kary Mullis in the 1980s. Or polymerase chain reaction, abbreviated as PCR, is a laboratory technique for rapidly producing, amplifying, millions to billions of copies of a specific segment of DNA, which can then be studied in greater detail. PCR involves using short synthetic DNA fragments called primers to select a segment of the genome to be amplified, and then multiple rounds of DNA synthesis to amplify that segment. Polymerase chain reaction is a powerful molecular biology technique used to amplify specific segments of DNA. Developed by Kary Mullis in 1983, PCR revolutionized the field of molecular biology by enabling the rapid and precise replication of DNA sequences. PCR is widely used in various applications, including genetic research, diagnostics, forensics, and biotechnology. Principle: the PCR process involves repeated cycles of DNA denaturation, primer annealing, and DNA extension. These cycles exponentially increase the number of DNA copies, leading to the amplification of the target sequence. How does PCR work? To perform PCR, the template is mixed with primers, polymerase, and nucleotides. The mixture is heated to 94 degrees Celsius to denature the DNA duplex, unzip it into single strands. Next, the sample is then cooled to 45 degrees C 60 degrees C, allowing the primers to base pair with the target DNA sequence, called annealing. Lastly, the temperature is raised to 72 degrees Celsius, the optimal temperature at which the polymerase will extend the primer to synthesize a new strand of DNA. Each PCR cycle, denaturation, annealing, extension, doubles the amount of the target sequence in less than 5 minutes. In order to produce enough DNA for analysis, 20 to 40 cycles may be required. What do I need to perform PCR? Template: the purified, double-stranded piece of DNA we want to copy. Primers: short synthetic DNA molecules that target a specific DNA sequence for amplification. taqDNA polymerase, a thermostable enzyme used to copy DNA. Free nucleotides, the building blocks of DNA. Thermal cycler or PCR machine, a specialized machine that rapidly heats and cools the samples. Components: 1 DNA template. The DNA containing the target sequence is to be amplified, or you may say template strand, or the new fragments of DNA that are made during PCR also serve as templates to which the DNA polymerase enzyme can attach and start making DNA. 2 primers. A primer, as related to genomics, is a short single-stranded DNA fragment used in certain laboratory techniques, such as the polymerase chain reaction. In the PCR method, a pair of primers hybridizes with the sample DNA and defines the region that will be amplified, resulting in millions and millions of copies in a very short time frame. Primers are also used in DNA sequencing and other experimental processes. 3 DNA polymerase. An enzyme that synthesizes a new DNA strand by adding nucleotides complementary to the template strand. Polymerase is the heat-stable, thermostable, DNA polymerase extracted from the thermophilic bacteria Thermus aquaticus. 4 nucleotides. The building blocks of DNA, A, T, C, G, are needed for DNA synthesis. Deoxynucleoside triphosphates, DNTPS. DNTPS consist of 4 basic nucleotides DATP, DCTP, DGTP, and DTTP as building blocks of new DNA strands. These 4 nucleotides are typically added to the PCR reaction in equimolar amounts for optimal base incorporation. 5 buffer solution. Provides the appropriate pH and salt conditions for the PCR reaction. PCR is carried out in a buffer that provides a suitable chemical environment for the activity of DNA polymerase. The buffer pH is usually between 8.0 and 9.5 and is often stabilized by tris-HCl. 6. Thermal cycler. A specialized machine that controls the temperature changes during each PCR cycle. The thermal cycler, also known as a thermocycler, PCR machine, or DNA amplifier, is a laboratory apparatus used to amplify segments of DNA via the polymerase chain reaction, PCR. The device has a thermal block with holes where tubes holding the PCR reaction mixtures can be inserted. Steps of a PCR cycle. 1. Denaturation. The DNA sample is heated to a high temperature, typically around 94 to 98 degrees Celsius, to separate the two DNA strands, breaking hydrogen bonds between base pairs. The first step in PCR is denaturation. Denaturation is required to separate the double-stranded DNA sample. It is done at 94 to 98 degrees Celsius for 20 to 30 seconds. It breaks the hydrogen bonds present between base pairs. 2. Annealing. The temperature is lowered to around 45 to 60 degrees Celsius. During this step, the primers bind, anneal, to their complementary sequences on the DNA template strands. The annealing step, 30 SEC to 1 minute, at temperatures 45 to 60 degrees Celsius, is required so that the primers bind to the complementary sequence on each of the DNA single strands. The primers are designed such that they bracket the target of interest, and the region of sequence that lies between them is referred to as the amplicon. 3. Extension. The temperature is raised to the optimal range for DNA polymerase, usually 72 degrees Celsius. DNA polymerase synthesizes a new DNA strand by adding nucleotides complementary to the template strands. Although this is commonly referred to as an extension step, a major purpose is to allow re-annealing of the PCR product into double-stranded DNA so it can be visualized using ethadiene bromide after gel electrophoresis or used for cloning. 4. Amplification process. After one cycle of denaturation, annealing, and extension, 2 DNA strands are created, each containing one of the original template strands. Subsequent cycles repeat these steps, resulting in an exponential increase in DNA copies. Each new DNA strand can then serve as a template in the following cycles. PCR applications. 1. Gene cloning. PCR can produce a large number of identical DNA fragments for insertion into plasmids or other vectors for cloning. 2. DNA sequencing. PCR can amplify DNA regions of interest before sequencing, allowing for more accurate sequencing of specific targets. 3. Diagnostics. PCR is used in medical diagnostics to detect DNA or RNA from pathogens, including viruses and bacteria. 4. Forensics. Minute quantities of DNA from crime scenes can be amplified using PCR for analysis. 5. Genetic research. PCR aids in the study of gene expression, genetic mutations, and gene regulation. 6. DNA mutagenesis. PCR-based techniques can introduce specific mutations into DNA sequences for functional studies. Variations of PCR. 1. Reverse transcription PCR, RT-PCR. Converts RNA to complementary DNA, cDNA, before amplification. 2. Quantitative PCR, qPCR. Measures the amount of DNA during amplification, enabling quantification of initial template concentration. 3. Nested PCR. Involves 2 sets of primers in successive PCR reactions to increase specificity. 4. Multiplex PCR. Amplifies multiple target sequences in a single reaction using multiple primer pairs. 5. Digital PCR. Quantifies DNA molecules by partitioning them into numerous individual PCR reactions. Polymerase chain reaction has transformed genetic research, diagnostics, and various other fields by making DNA amplification rapid, efficient, and highly specific. Its versatility and adaptability have contributed significantly to advances in molecular biology and biotechnology. For detailed information on complete PCR reactions in step-by-step, stay with Dr. Netra Pal Sharma.